Okay, thank you, Damai, for sharing the screen. For this team, I will present our project. So, hello everyone. I'm Alvaro as the one of the member of Beta Indonesia team, and in this project, we want to introduce X-Zymes. So our biofoundry name will be X-Zymes, which is planning for accelerating research regarding on the extremophiles from Indonesia to find and produce the potential protein and also any other compounds. So next slide. Well, this is our table of content of our presentation today. There will be background and our idea and also our plan. Next. Okay, as the background, we all know that extremophiles are the treasure trove of novel biologically active molecules with unique properties or many proteins that also have unique properties that can be used for a wide range of biotechnological processes, notably those of required extreme temperature, extreme pH, salinity, and the pressure for efficient running in the most cost-effective manner. Indonesia, as the country which is located in the Ring of Fire region, has enormous amount of diverse extremophiles, which is highly potent for various kind of utilization. However, up to date, the isolation and identification, also utilization of these extremophiles are still minimal. So a group named the Indonesian Center for Extremophile Bioresources and Biotechnology, or ICEBB, which focus on extremophile culture collection, only have approximately 270 collections, while there are still huge potential of diverse strains to be explored. Therefore, we want to propose X-Zymes to cater extremophiles identification and its potential genome production, potential protein production to be used in various sectors. So next. Yeah, to conduct our project, we firstly would obtain the extremophile bacterial strain from the customer or site of extractions. Then we will proceed to extract the DNA and sequence them with PDQEX and Minion, which this method only has already used for another cases in Africa. So PDQEX is the method of extraction, and Minion is used to do the sequencing. Then we could upload the sequence into the GetExtreme. So GetExtreme is bioinformatics genome mining platform, which is developed by one of our member from uh, University of Technology Sumbawa, WANA, with collaboration with in collaboration with ICEBB, and we are going to collaborate with GetExtreme we use it to detect potential gene which allows this bacteria to sustain in extreme conditions and also can be used for further biotechnology application. So after we obtain the potential gene, we can give the result and offer the further production with the customer. From there, we will design the optimum cases for desired enzyme or compound production. And because one of our member is one of IGEM team from Brawijaya University, we will have the collaboration with IGEM Brawijaya for the cases designing, because the project is design a customable and optimum strain of E. coli that can produce extracellular protein with good expression quality. So after that, we are going to do the cloning and transformation of methods, such as using the multiple electroporator that has been also developed by one of our member from Example University. Then we are going to produce the gene of interest, whether it's enzyme or maybe integrate maybe another compound that potential to be used. So this scheme is actually we will do this scheme when we have a small demand. This is our early phase of biofoundry. So we realized that we are still using like DIY equipments that have been developed by our members, and also that get inspired by Enzyme from last presentation. But for the next plan, we are going to have collaboration with Indonesian Center for Science, because I've been doing an internship there for 3 months, helping startups collaborations which need technologies, like high throughput technologies like a modern liquid handler and many more. So if we want to scale up, we will have collaboration with that research center. Because I also know the procedure, I think it will be easier to have collaboration. And for further plan, we are going to, of course, we want to have our own equipments and instruments. But for our first step is doing by the frugal concept. And for the next, we are going to do with collaboration concepts. And lastly, we are going to run independently. So next slide. Okay, so as the concluding remarks, from these methods, we can obtain several things. Firstly, we can enrich the extremophile strain collection, especially for the variants found in Indonesia. And also by doing this, we increase the diversity of potential extremophile to be utilized in various sectors. Secondly, we also provide, and third, we also provide the identification service for extremophile in Indonesia, where we can do this lot of potential. We can maximize the utilization of extremophile strains, and furthermore, we hope to bridge and accelerate the research process into its industrialization, because we believe one cycle that said the productivity of research can increase the productivity of industry and also can increase the productivity of economy in Indonesia. So I think that's all from Beta Indonesia team. Thank you for the opportunity.
Thank you very much. That's very interesting. Extremophiles, I don't see a lot of people working with those. So now we have opening for questions, and we'll do 5 minutes.
Question for you. What sort of equipment do you need for this pipeline, and what do you have already?
Okay. Can I answer directly? Yeah, okay, so actually we have list our equipments, but we cannot show in the presentation because we have limited of time. But so for this timeline, I will explain a little bit. We are getting the sample from the customer, and we have to do the DNA extraction with PDQX. This is like a machine to do the DNA extraction that has been used by I forget the name of the researcher, but this is really fast. We can extract the DNA within 20 minutes instead of 3 hours. She said in her presentation. And also we are going to use Minayon because we know that Minayon is really handly and also simple for the sequencing. So this is the first two equipments that we use. And then we are going to use the software that has been developed by our members called GetExtreme. This is a genome mining platform which database contains of many information from extremophiles in many conditions, living conditions. And also for the design, we are going to collaboration with Agent Brawijaya. So here we also provide the our methods for do the genome editing. So we have to identify one gene that can increase the production for extracellular protein. We are collaborating with IGEM. For the DIY equipment method, we also using one of our member developed like portable electrophorator. Like, so if you know the electropen, we are following the concept of electropen for the transformation, for example. We have tested it in agrobacterium, so I think it's going to be potential for E. coli as well. And next for the clone, that one is for the cloning and transformation. So for the protocol, we are going to use like microplate reader and also what we call it, the handler, liquid handler, but it is only for the next scale production. So just as I explained, for the first, we are going to use the DIY methods and also DIY protocols. We also have, collected the protocols from like the Odin, like something like that. We also collected an easy protocol and also low cost protocols. And for the next step, we are going to collaborate with research center here because I have been interested there to help startups. So I know that they have liquid handlers and also advanced instruments that we can use. So instead of buying the instrument, we want to first do the collaboration. When we have enough capital and funding, we will buy our own for the production, for the bigger production. Am I answering the question?
Yep, that's good.
All right, any other questions from other teams?
Sorry, may I ask about what the database looks like? Just curious, because like in my final year project, I kind of make the similar stuff like that as well for Extremophiles. Like, I just want to suggest that maybe like creating a map like for the location of the Extremophiles that you found might be helpful. Like maybe you can say like, so like maybe in this particular lake, like they produce like a lot of sulfur bacteria or something like that.
Okay, so the get extreme. You mean the get extreme, right?
Yeah. I'll share the link if you guys want to check maybe.
So actually, get extreme is okay. Get extreme is it's similar to anti-smash. Do you have you ever heard about anti-smash, which is a genome mining platform for bacteria or any other prokaryotes? Actually, Wana is here, so he is developing this with his team and collaboration with ICEBB. So the method, the concept is like the anti-smash platform. So we are input the genome sequence, and then the platform will search based on the database, and then the result will offer us the potential genes that could be used for biotechnology application.
I see. Can you share the link in the chat, please? I want to check it out actually. Thank you.
Oh, okay. Warna, can you help me? Or maybe the platform is still being developed. It's not finished yet. Can you?
The
Can Warna
platform
help?
is it's developed and
Also, it is still being developed. Maybe
It's okay.
you can check it on the Instagram of synbio.id because the project is being contested in BIOS competition.
Oh, cool. Okay, then I'll check it out later. Thank you.
Okay.
All right,
Anyone
awesome.
any questions?
Yes.
So I just
Extra
had
question.
one question. Yeah. Okay. So what I don't really understand clearly is who will be the most affected because of this problem and the solutions that you guys are proposing? Like, what is the need for this particular usage of extremophiles? Or where can it be used?
I'm sorry, can you repeat the question?
So who will be most affected by this extremophile solution that you're presenting, and how else can you use this particular project?
Oh, okay. Thank you for the question. Actually, many researchers in Indonesia is now start to interested in extremophiles. So in Indonesian Center for Science, for example, they have the division for microbiology, and they are developing new drug discovery and novel protein from this extremophile. So that's one of the example. And also ICEBB is also has so many like they have grow in many region of Indonesia, and they also conducting research, but we want to accelerate the researchers so the result can directly being produced with a frugal approach, and we are going to introduce to the industry which needs the enzymes. So actually the plan is accelerating research process, and we help the industrialization, because I think I know that it will be we will need the permission from the government, but for the first step or any smaller scale, we are helping them. We have found that also Indonesia's researcher also doing research on extremophiles, but I think the output is still not uh, should not be only publication. They will also have to introduce to the industry. So we see the potential. So we are bringing this topic.
Okay, sounds interesting. So